Namaskar everyone. I am Pihu Gaur, a student of India International School, and today the topic of my presentation is AI generated synthetic genes for investigating novel biomarkers of systemic lupus erythematosus. So first, let's understand what we mean by systemic lupus erythematosus. SLE is a complex autoimmune disorder in which one's own immunity system attacks their own tissues. It has diverse clinical symptoms. The most noticeable symptom is the butterfly rash that appears on the cheekbones and nose bridge of a person. It mainly affects women aged between 15 to 45 and poses significant health challenges. Timely identification and intervention are crucial for effective SLE management. The main challenges with SLE study is that it does not have robust research material and effective mechanism. There are mainly three main causes of SLE. The biggest cause is the genetic predisposition of an individual. So, many a times, a person that is diagnosed with SLE tends to have a family history of this disease. The genetic composition paired with the environmental influences may cause them to have SLE symptoms. Hormonal factors also play an important role in this. So the main question that arises is how can we prevent SLE? We are preventing SLE through genomics and machine learning. So as we have discussed earlier, some genes exhibit a latent risk like TNIP1 or TNFAIP3, and their early identification helps in preventing SLE. Genomics, which is a branch of omics, has significantly contributed to the accumulation of large-scale data, which fuel the investigation further. Machine learning has enabled the integration and analysis of omics data, leading to the discovery of new biomarkers with potential application in disease prediction, patient stratification, and precise and personalized medication. So we need to investigate additional genes in order to understand SLE as we have limited biomarkers. The research problem is investigating novel biomarkers of SLE and generating synthetic genes using generative models of machine learning. So before moving further, let's first understand machine learning. Machine learning is a subfield of artificial intelligence, and there is a subset of machine learning, which is deep learning, in which comes the generative AI models. And generative AI creates content autonomously and generates diverse outputs creatively. The deep generative models can be classified into many categories on the basis of maximum likelihood. From all the given models, genomics mainly uses the generative adversarial network and variational autoencoder network. And we have used the variational autoencoder for our research purpose. And generative AI in omics have been employed to create artificial protein and drug molecules. So when it comes to generative AI in SLE study, in 2019, Periasamy and Byred conducted a phenotype study in which they utilized the CAN model technology, which is part of a deep generative AI, and analyzed images of butterfly rashes, the symptom that we have discussed earlier, to better associated with SLE. But till now, there has been no evidence of the application of such technique when it comes to the genomics of SLE. So the main aim of our project, of our present research, is to design a prototype based on the generative model to generate synthetic genes using existing ones. So there are mainly three steps in our methodology. The first is that we identified 8 genes that are known as the key contributors to SLE in both Asian and European populations. In the second, we got this gene sequences from UniProt. The sequence length varies from 200 to 4,000 in the dataset. In the third step, we had the VAE model, which was implemented in Python on the Google Colab platform. The study made use of essential libraries, including TensorFlow, Pandas, and Keras. So why have we used variational autoencoder? So variational autoencoder uses two neural networks, which is the encoder and decoder, and it allows unlike other models, it allows controlled generation of data, enables uncertainty modeling, and simplifies optimization and model convergence. So for the working of a VAE in our system, we had taken gene sequences from the UniProt. We first tokenized and encoded it into a trained data set, in which later we fed it to the encoder as an input. The encoder compressed the given input into a latent space, which was further given to the decoder, which we decompressed it and we got a synthetic gene sequence. Both the encoder and decoder were created using recurrent neural network. This was the architecture of our encoder and decoder. So in the end, we successfully generated 5 synthetic gene sequences. These synthetic genes can be subjected to further examinations as they may present a valuable opportunity for conducting comprehensive studies. They may lead to the discovery of novel biomarkers and facilitate the exploration of their interactions with pharmaceutical compounds. These were the references that I have used, and thank you.